And thank you everyone for coming. It's really wonderful to be here today to talk about how life changes planets and how that relates to ecological complexity. Faced with this title, I decided to view it very much in terms of what is the ecology and history of life on Earth that I would like essentially everyone to know regardless of their background, so astronomers and physicists and chemists. So that's the viewpoint, which means that probably there'll be some stuff maybe for some of you that's quite basic, but hopefully everyone will be able to follow along. So let's see. So when we think about life, especially as a biologist, we tend to think in terms of the details. So what's going on in cells, for example, or why do animals behave the way they do? But if we're starting to think about whether life exists outside our own Earth, we can't really focus on these relatively small details because with the potential exception of sample return from Mars, we'd never be able to detect these really, really small things. So instead, we need to be thinking about things differently from a planetary scale. And in order to detect what life might look on other planets, we need to understand the history of life on Earth. So when we're thinking about the evolution of life on Earth, we tend to think about in terms of the major transitions that life undertook, because that provides a framework for then going on to think about things like the probability of such events occurring and what indeed drives such changes. So Maynard Smith and Zara Murray wrote a book which laid out what they considered to be the major transitions. So these start off with the formation of molecules capable of replicating, which then assemble into chromosomes, i.e. RNA. These then change to form DNA. So that's proteins as enzymes, where enzymes are the molecules which act as catalysts to form specific biochemical reactions. These are then parceled up as prokaryotes, that is single-celled organisms in which DNA is contained. The next big step is eukaryotes. So that's where the genetic material is kept within the nucleus. Then comes sexual reproduction, which brings with it many key benefits for evolutionary processes, such as genetic variation in offspring, so the ability to select, otherwise known as survival of the fittest. After sex comes multicellularity, such as we see in animals and plants and fungi. Then we have eusociality, which is the ability of individual organisms to prioritize the survival of their population over their own. This includes things like worker ants in big colonies, and also naked mole rats, where only one female breeds and most of the colony do not. The final step in evolution is social cultural evolution, which is humans, whereby culture changes, such as beliefs or the skills, and these changes have similarities and also differences with genetic evolution. However, these transitions are much debated because there are some very clear problems with them. For example, the evolution of eukaryotes and sex are very much interlinked. Prokaryotes reproduce asexually, whereas kind of the major groups of eukaryotes, that's animals, plants, and fungi, can reproduce both sexually and asexually. You also run into problems with some of these stages when transitions have evolved independently multiple times, such as eusociality and multicellularity. It then stops making so much sense when trying to build up these sorts of models. If you have multiple transitions, which one do you choose and how do you decide to go about it? So if we think about the evolution of multicellularity, okay. So here we have a phylogenetic tree describing the relationships of different organisms. This is a bit of a funny one because it's very plant focused. You can see lots of different photosynthetic groups. We've got algae, land plants, phytoplankton, and all of animals are over here encapsulated by this little red dot. But what it makes very clear is that multicellularity has evolved at least once in every eukaryotic clade, and numerous times across the prokaryotes as well, repeatedly for things like the green algae. Which means that it's reasonable when we're thinking about the major transitions of life on Earth, not really to consider multicellularity necessarily the key one, but to try to think about other ways and other transitions that we should be focusing on. Okay. Sorry. So another massive shortcoming of the traditional major transitions in evolution is a lack of photosynthesis. It's not explicitly listed, despite how important it clearly is to the world we live in today. But it does run into similar problems to multicellularity, in that it's evolved multiple different times. So chlorophyll-based photosynthesis has only been found in bacteria and eukaryotes, so none in archaea. But within bacteria, you have many different sorts of photosynthesizing bacteria. So things like cyanobacteria, purple bacteria, and slightly weirder ones like filamentous anoxic phototrophic bacteria, or green non-sulfur bacteria. And they vary a lot from being broadly phototrophic, such as cyanobacteria, but mostly not, such as with protobacteria. However, organic photosynthesis has only evolved once, and it's that once that's really, really key to the evolution of life on Earth. So when we are thinking about major transitions in terms, we run into some of these big problems. How do we distinguish the very important transitions, such as the multicellularity of animals versus that of slime birds, for example? And one way to do that is to not consider the relative biological complexity, but instead consider the ecological complexity. That is, what are the transitions that have major impacts from life onto this planet? So let's look at the history of life, not through a biological lens, but through an ecological one. What are the key innovations that changed the biosphere on a planetary level? So the four that I'm going to focus on today: the origins of life, the evolution of oxygenic photosynthesis, which led to what is known as a great oxygenation event, which is where we see a really massive uptick in the amount of oxygen within the biosphere. The evolution of animals, which is when we get another increase in atmospheric oxygen. And finally, the colonization of land by animals and land plants. Now, it wouldn't be unreasonable to suggest that humans and the damage that we have wrought on this planet could be a fifth one. However, I'm very much going to leave that for now, because while humans made a very large impact on the biosphere, I suspect that detecting other technological species on different planets would be much, much easier than those of non-technological species, such as this. So, point of order, since you've got a very mixed audience, in Earth sciences, geology, paleontology, 0 is where we are now. And so the Earth formed about 4.5 billion years ago. I noticed chatting to astronomers that they do it the other way around. So 0 for them is when the Earth formed. It can get very confusing if you're talking about scales and you're talking about the different things. So from what I can gather, most of the people here today are from the School of Physical Sciences. So I wanted to take a few moments to go through what exactly I mean when I talk about ecological complexity, and indeed why it matters in a planetary context. So starting with a very basic question, what is ecology? Most of us have a sense of this. It's how living things interact with the world around them. But there are many subdisciplines within ecology, not unlike the biological sciences. So in biology, we have genetics, physiology, and zoology, for example. And you can group ecology in a similar way. Some people do it by ecosystem type, so marine ecology, terrestrial ecology, microbial ecology. But I prefer to think about it in terms of the levels of the organism upwards, because different methods and different questions are asked depending on whether you're looking at an organism or whether you're looking at a population. And the same's not true if you're comparing terrestrial and marine biology, where a lot of the same methods can be applied. So if we start with organism ecology. Now, this organism ecology are things like behavioral ecology. So why do antelope herd, for example? But also things like functional ecology. And functional ecology is one that we come up most in paleontology, for example. So here we've got an early tetrapod skull, and they analysed the sutures in the skull. And what they determined is that, looking at different forces that act upon it, that it fed mostly in water, but it was also capable of hunting a bit on land. It was adapting a bit to land. And this is definitely functional ecology. It's looking at the function of the form in order to infer the ecological interactions. We also have population or community ecology, which is where we're talking about competition between species or even within a population, and also things like predator and prey dynamics. Things like landscape ecology, biogeography. So how are species distributed around the globe, for example, and what's driving the different patterns that we see? The other branch is theoretical ecology. And this is what is driving ecosystem structure, what is making ecosystems function the way they do. And how does that relate to things like ecological resistance and stability over long periods of time? And it's very much, when I talk about ecology, it's very much talking about it in terms of ecosystem structure and function and stability. So more a theoretical ecology approach than, for example, organism ecology. So if we move on to ecosystem complexity, again, I would say most of us have a reasonable sense of what it is. A tropical forest is relatively complex with all of its species, various different kingdoms. You've got your fungi, you've got your plants, you've got your animals, and they're all working together and living pretty happily within that world. In contrast, something like a farming monoculture isn't very complex. It doesn't have very many species. But why does ecosystem complexity even matter? Well, it matters because while more complex ecosystems are more stable. That means that they're much better at adjusting to changes and so surviving over long periods of time. So if we're in a rainforest and a species tree, for example, gets wiped out by a pathogen, for example, then others can fill its role in terms of habitat or food. However, a crop pest can easily wipe out an entire field of the crop. So understanding ecological complexity is very much a key area of research for modern ecologists. And a large part of this is something known as the complexity stability debate. So we know from our own experiences that more complex ecosystems are more stable. However, if we build up very simple models, so that species with interactions between them, actually more complex models are less stable. If you take out a species, the whole thing is much more likely to collapse. So this problem was first articulated by Bob May in Oxford in the 1980s. And since then, there's been a huge amount of work trying to understand what the differences between real and models ecosystems. And it comes down to a few different factors. And so it's about the patterning of the links between all the different species in the ecosystem, but also the strength. So if you have too much of a food source, for example, that will massively increase, but that means the predators above it will have more to eat, so they will increase. But as they increase, they then need more food, and so then will then eat the kind of overabundant prey species. And so you get this feedback loop, and the system is capable of correcting itself. So hopefully now everyone's got a reasonable sense of what I mean when I talk about ecological complexity. So it means we can now start thinking about how ecological complexity has changed here on Earth and how these changes feed into the biosphere changes that we can see, such as with these different phases of ecological evolution on Earth. So because our motivation for thinking about these things is very much in terms of detecting life on other planets, the key question is what is possible. So clearly detecting prokaryotic from eukaryotic cells will never be possible remotely. And that indeed is why we're thinking about it in these very, very broad terms. So I'm not going to talk a lot about biospheres, biosignatures, that is signs of life on other planets, for two reasons. Firstly, because you could easily spend a lecture or two or entire lecture series talking about different sorts of biosignatures and how you distinguish them from abiotic signals. But also, I'm not going to go into too much detail because that's not really what I like to focus on. I'm more interested in the actual ecology and ecology. But I am going to just briefly discuss how these four different phases of ecological complexity might manifest themselves on other planets. So the key tool we have for detecting life on exoplanets is the use of transmission spectrums of exoplanet atmospheres. And the idea that different molecules have different radius of absorption. So by analyzing the spectrum, we can work out what molecules are present in the atmosphere of different exoplanets. So the use of oxygen as a biosignature is very, very common because it's pretty unlikely, but not impossible, to have it in large quantities in the atmosphere without any kind of biogenic generation. However, a large part, because it's not a sure thing, needs to be taken into consideration with other different combinations of molecules, such as methane and water. But how would all this apply to different phases of life on Earth? Well, this is all very good in theory, but would we actually be able to detect life on Earth if we were aliens looking back at it with the same sort of measuring devices. So this rather nice theoretical study that Paul Rimmer put me on to looks at exactly this question. So including 5 different phases of life on Earth. So they've got 3.9 billion years ago. So before we have oxygenic photosynthesis, then we have 3.5, which is around when we think it first originated, but we still have basically an anoxic Earth. And then we've got the 3 different phases in the oxygen levels in the Earth's atmosphere. And what we can see is it changes, well, both in terms of methane levels. You see very dramatic differences between the anoxic and oxic phases, and also in terms of oxygen nitrates. And we can see again the different spectrums for the oxic phases. So what the take-home point of this slide is very much we can apply these methods to Earth, but really we're not going to be getting strong signatures till about 2 billion years ago. So the other aspect that wasn't really covered by the previous study was how we'd go about detecting life on land in terms of land plants. And this was one study that was quite nice in that it was talking about how infrared signals would change in the presence of plants versus other similarly colored rocks. So things like green leaves, they have a lot more reflectance than similarly colored abiotic variables, as well as less polarization. So these are just a very, very short glimpse into biosignatures, but I think we can all agree that in theory it should be possible to detect these different phases in Earth's evolution. So let's start to dig into what was actually going on planet Earth, starting with the origins of life. So it's very, very hard to detect life right at the start. You've got massive problems with pseudofossils. And this, of course, also applies to sample return from Mars. We think it's quite likely that life started relatively quickly during the first few 100 million years, but it's really hard to know for sure. And there are many different suggestions of possible first life on Earth. So things include these ionoxic tubules here, which are kind of 4.1 to 3.7 billion years ago in hydrothermal chert from northeast Canada. And they were interpreted as the mineralized sheaths of iron-oxidizing bacteria, and hence the oldest fossil organisms. However, it's been shown that most of these features, the very simple microstructures, can be reproduced in a lab kind of chemical garden, which of course may have natural analogues in hydrothermal vents. We've also got the potential 3.8 billion-year-old stromatolite. But again, this doesn't bear under kind of more analysis, because if you look at the 3-dimensional shapes, they actually show that they're more likely to be a deformation or alteration of other structures. We also have a very recent paper that came out, which was suggesting that these filamentous structures were fossils from about 4.28 billion years ago, due to their mineral associations and their resemblance to younger filamentous fossils. But again, they're kind of they don't really bear up under more scrutiny. So what the kind of take-home message is, like, if we're looking for actual fossil evidence right around 4 billion, it's really, really hard to distinguish things that could be reasonably abiotically generated versus actual fossils. But why do we think there was life at this time? Well, in part because of much higher methane levels around this time, which the lower limits given by this here, which is mass omissions from sulfur. So increased methane, likely generated by methanegens, coupled to molecular clocks, which is here. So this is a nice molecular clock. Generally, you have to be cautious about them, but this is for archaea, and looking at horizontal gene transfer to try and work out when they might have likely originated. You may notice that it goes, confidence intervals go back beyond the origins of the Earth. But nonetheless, we know that there was liquid water from oxygen isotopes and zircons right around 4 billion years ago. And how would we get this if we actually have a fainter sun? So the sun was 25 to 30% weaker than it was today. And it's likely, or thought, that greenhouse gases such as methane were likely present in large numbers. And this was enough to enable us to have a kind of liquid water on this planet. But how would we think about this in terms of existence here on Earth? So if we do have life very, very soon, and then they persist for a very long time, when we're thinking about the first life on Earth, there's often this kind of assumption that they would have occupied a very small ecological niche. There's this tiny little pocket where all the conditions are right that life has to initiate from. So one of the suggestions is hydrothermal vents, but it could indeed be any other sort of patchy abiotic habitat that's particularly conducive for life on this planet. But this means they're very spatially limited, but also temporally limited. So, for example, with hydrothermal vents, some can last up for 10,000 years, but a lot will only live very, very quickly for 2-3 years. But we still have a continued existence of hydrothermal vent communities. And that's because they can disperse very, very well. I've got very good and long-lived dispersal mechanisms, but it's also because of the existence of islands. So you can have kind of, I guess, larvae of different creatures kind of jumping between the different vent zones, but also things like whale falls. So the sulfur in the whale's bones actually provide little islands for hydrothermal vent community animals to kind of jump to if there aren't enough hydrothermal vents close by for them to survive. But it's all a very precarious balance. So in terms of ecological stability, these are not particularly stable systems, but they maintain because there are enough of them. Another big problem with these very small ecological niches, especially if they're patchy, is the problems of fragmentation. So, because small populations are much, much more vulnerable to extinction than large ones. So there's a particularly sad case of a Thai guinea fowl population that was nearing extinction and that was being monitored very carefully. There were 4 of them left, 1 male, 3 females. The male got hit by a falling tree and died, but that essentially doomed the entire wild population to extinction because of this chance event. If it had been one of the female ones, they would have been fine. Obviously, humans intervened and, the population still exists. But if you have a lot of very, very small little communities, you're much, much more vulnerable. The fact that we had life and it continued for hundreds of millions of years means that there was enough of it around. So it must have been extremely abundant and extremely well distributed in space and time. So spread rather quickly rather than remained very local. So moving on to oxygenic photosynthesis. Around about, we start seeing the great oxygenation event about 2.5 billion years ago. Now this is important in on itself, but it's also important because cyanobacteria are the first proper, cannot argue with, evidence of life on Earth. So here we've got some stromatolites from 3.5 billion years ago in Australia. We can see them here. So these are mounds of sediment that are formed by cyanobacteria, and they form kind of biofilms that bind and cement grains of sediment together in progressive layers. So most of life on Earth was dominated by the stromatolites for literally billions of years. They are still around today. So here's some from Shark's Bay in Australia. You can see the little mounds, and there are some in the Bahamas. But they're really, really rare now because they need a long time to form, and so they can't be disturbed by fish or other creatures digging in the sediment. They exist in their thousands in Shark's Bay because the water is particularly saline, so that more animals can't survive there. So we've got our oldest evidence of life on Earth. We also have microfossils from about 3.2 billion. So these are tiny little fossils that you find by dissolving rock in hydrofluoric acid, and you get them thawed out. And it's microfossils that mean that we have a really good documentation of the evolution of life on Earth from 3.5 billion years or so, I guess 3.2, to when we can start seeing them properly in the fossil record. So the introduction of these oxygen-producing cyanobacteria led to the buildup of oxygen in the atmosphere, accumulating in about 2.5 billion years ago, where we see oxygen rise during the great oxygenation event. So this changed from kind of almost no oxygen to about 100 times, 1/100th of the current atmospheric levels. Now we can't directly measure this in the geological record, and so it's detected in quite a few different ways. So one of the ones that's most popular is the use of isotopes, particularly sulfur and mass-independent fractionation of sulfur, which detects the presence of sulfur. The presence of it shows that there was some photodissociation of sulfur, where the sulfur molecules is broken down by UV radiation. And this breakdown could not occur if there's an ozone layer that protected the lower atmosphere from UV radiation. And we can see here the differences that corresponds to that. And so there's a massive change. And it's not just sulfur, there are lots of other isotopes, not other different methods that back up this consistent pattern. So the time period between the end of the Great Oxygenation event at about 1.5 billion and the origins of animals around 800 to 600 million years ago is sometimes particularly harshly referred to as a boring billion. Because you have relative tectonic stability, limited climatic changes, and perceived slow biological evolution. So life at this time was still very much dominated by cyanobacteria. But it's around the start of the boring billion that we start to see eukaryotes first evolve. So the most definitive ones come at about 1.6 years. We've got the fungi. These are both Nick's fossils, but he's had enough. And we know they're eukaryotic because they've got these very complex protrusions coming out of them. And you simply can't get that sort of level of complexity with prokaryotes without fully motorized cytoskeleton and endomembrane system. Then at about 1 billion years ago, we get the first evidence of sexual reproduction in the form of the red algae Plangiamorpha, which is where we see differentiated reproductive cells. And so it's the oldest evidence of sexual reproduction. In terms of ecological complexity, the vast majority of life on Earth was still using lights because it was cyanobacteria, although there were some heterotrophic fungi and bacteria. So very kind of limited in terms of complexity. So now moving on to animals. I could quite easily spend a lot of lectures talking about the importance of animals and the ecological complexity and how the ecology is driving the evolution, because that's what my research is, but I'm going to try to be a bit restrained. So around about 600 million years ago, we start seeing large complex fossils appear in the rock record. And this is during the Ediacaran time period. So a lot of them have anatomies that are only found in the Ediacaran. So you can see here, this is China. We've got branches that are branches of branches, and this is called fractal style branching. You can get up to 5 different levels of it. And so while they look superficially like plants, because they're found in very deep water rocks, we know that they can't have been photosynthetic. But from recent work by Frankie Dunn in Oxford and her colleagues, we know that they are indeed the first animals. But they didn't really do much in terms of interacting with their environment. They weren't affected by it, and they didn't interact much with each other or with their local habitat. So we need to move on a little bit into slightly younger Ediacaran animals, which include ones that are starting to look a lot more similar to our modern animals. So we've got Sphagina here with its segmented body plan, and it's bilaterally symmetric. We've got Dickinsonia here, which admittedly controversial biomarker work has shown that steroids are consistent with metazoans, so animals, but not with bacteria or fungi or anything else. We also have Kimberella that we'll be hearing about in a little bit too. And this is suggested to be a mollusk-like creature because of the way it scratches up the sediment in a similar way to snails. We also have Hutia here, so one of Alex Liu's fossils that's thought to be a stalked jellyfish with potential muscle fibers. And then we've got one of my favorites, Thectardus. If you're wondering, it's just a little triangle, Emily. It's like, yeah, it is just a little triangle. But it is important because it's been suggested to be the oldest sponge. So one of the simplest animals there are, this little triangular Thectardus. But the really, really big difference in the innovation of Ediacarans was their ability to move around. And by moving around, they're changing the environment around them. So here we've got Yorgia, and you can see it's got body prints behind it, it's got footprints. So there's one little trail coming along here. And we've also got Kimberella up here, some down here. You can see the scratch marks where they are scraping away at the sediment in order to feed. And this is a really, really big change from slightly older Ediacarans and also things like macroalgae, for example, because by doing things like moving and feeding, they're changing their environment very, very quickly. And what's interesting is that these changes in the small scale habitat are actually thought to then provide more selection pressure and drive more animal evolution, particularly of bilaterians. So the other really big new animal thing is the evolution of biomineralization. So that is the ability to build skeletons such as shells and bones. So this is really massively crucial since reef building organisms are what are known as ecosystem engineers, because they change the local environment, but beyond, well beyond that of their own lifetime. And we know today reefs are very much biodiversity hotspots because they provide new habitats for other organisms. And they provide places not only for them to anchor to, but also to shelter from predators, for example, as well as providing food. We very much are aware of how precious these reefs are and fundamental to the functioning of the marine biome because of all these different ways in which they change the local environment. This is the Cloudina, which is the first, one of the first reef forms here. And in this Cloudina, there have been, you find the first evidence of predation just before you get to the Cambrian in the form of little boreholes, which link up to where you'd expect the body of the organism to be. But little Cloudina boreholes aren't really proper predators, but they do explain why we need a hard shell. We need a hard shell in order to protect ourselves from predators. So once we get to the Cambrian, we start to see predation really, really take off. So the largest is this Anonnocaris. It was up to 60 centimeters long, but a nice reconstruction by people here at the ROM. And it's thought that a lot of the diversification of early animals in the Cambrian was a predator-prey arms race. The predators got bigger kind of hunting devices, and then the trilobites had to adapt and get more spines, for example. But what's really crucial for us is that predators are really, really important in terms of the stability of ecosystems. We're very much aware that if too many apex predators get killed by humans, then the ecosystems often spiral out of control. So, for example, sea otters eat sea urchins in kelp forests. This is a healthy one. This is, however, if you kill too many sea otters, the sea urchins start to take over because they eat all the kelp roots. And if you really don't get your otters in time, then you actually destroy the entire ecosystem. It all disappears, and it's very, very hard to get back. But there's also less catastrophic effects of predation. So we've got some really nice, very early community ecology looking at starfish predation on rocky shores. So starfish, like a lot of predators, will prey switch. They will eat whatever is closest and most abundant. And this is actually really useful because what happens is when they've eaten too much of one thing, they'll then switch to the other one. So for example, they'll be switching between the two different groups here. And what this means is you're maintaining your diversity. Without the starfish, whatever is a better predator would quickly take over. And you're left often with just huge areas with this one or two species rather than more diverse communities because the predators are exerting a top-down effect on the ecosystem. And what's really interesting, I think, in terms of kind of planetary scales of ecological complexity, is what you're seeing here with, to some extent with the reefs, but very much with the predators, is you're having a biological stabilization mechanisms. You're controlling your abundances, not because of your resources, your environment, abiotic sources, but actually the biology is doing that for himself. But the evolution of predation is very, very closely linked to the evolution of free swimming. So there is evidence of scavenging in the Ediacaran and one or two potentially pelagic species, but you don't get proper pelagic predators to the Cambrian. And this is where the massive global impact of swimming animals really shows itself. Now, these are ideas that were developed by Nick Butterfield, but we can see here the effects of swimming animals. So this is acoustic data from the sea. Animals will reflect back acoustic signals more than water, and that's the red. And you can see that at night you have a lot of animals towards the top of the water, but not, and they kind of sink down during the day. And the reason they do this is because they don't want to be eaten. So they will hide away from the light. But the net effect of that is you're mixing up the water column. So before we get things that can swim around, you have quite a stratified ocean. You don't have much mixing. You do have plankton, prokaryotic plankton, but it doesn't mix the ocean in quite the same way that eukaryotic plankton does. But once you get things like predators and other things that are moving around, you start to get mixing up of the ocean. And this is what's causing the increase in atmospheric oxygen that we see. At least this is what Nick's saying. And so it's very, very much crucial to the evolution of early animals. So animals do fundamentally change the ecological complexity on Earth. So they do it through their swimming. They also do it through burying in the substrate. So as you bury further down, you get oxygenation of the sediment. What's quite interesting is we've seen some nice food web analyses that show that pretty much the entire trophic system that we have in our modern oceans was established right back in the Cambrian from analyses of the Burgess shell. So we flip in a very short time between having very kind of simple systems to very complex systems that are pretty much what we've got today. So the final transition I'm going to talk to you about is of land plants. So that's about 480 million years ago. So we start off with non-vascular plants, so things like mosses and liverworts, and then we get the vascular plants coming in about 100 or so million years later. So what's the first evidence of land plants comes not really in the form of body fossils, but in the form of these spores. And these spores are a bit kind of an intermediate morphology between things that we know definitely are land plants and earlier forms which are rather uncertain. But then, and so this is the oldest evidence, about 480. 430 we get Cookinsonia, which is here, and it's in its spore producing phase, so it will then produce spores. But some of the most really incredible fossils of early land plants come from the Rhynie Chert in Scotland about 410 million years ago. So these were preserved in incredible detail down to the cellular level because of their proximity to a hot spring, which led to very early diagenetic silica perimineralization. So the communities contain both non-vascular plants, like giant mosses, as well as vascular plants, and some are up to 20 centimeters long. And you can see, I mean, these are literally, so these are fossils, fossil plants over 400 million years ago, and you can see the cells. You can see all the details. I mean, I find this absolutely amazing. And you also have arthropods, so you've got some nematodes and arachnids here. Absolutely, I think it's just barely a millimeter big. Absolutely amazing level of detail. But you don't have roots or leaves of the kind that we're familiar with today. So the oldest kind of modern style tree, if you will, is found towards the end of the Devonian, around 370 million years ago. And Archaeopteris is one of them. And it has things like wooden trunks and leaves and seeds. Well, these are actually really, really important in the history of life on Earth, but also in the power that it gives the trees to then affect the biosphere. So wood is crucial for enabling structures to grow and reinforcing very large size. And some of these Devonian trees were up to 30 meters tall. You also see the first proper leaves. Leaves are really, really novel because it means that per unit biomass, you're collecting much greater amount of energy for your area. And so this is kind of massively transforming the physiological capacity in terms of energy capture, but also energy efficiency. So the impact that each organism has on its environment. And finally, and very importantly, mustn't forget about seeds. Now, seeds really do start to mix things up. So prior to seeds, land plants had to rely on water to carry their spores into new areas. And seeds are a very big improvement on spores because they can be transferred when dry, but also that the embryo can stay dormant until the environmental conditions are suitable for seeding. So these seeds meant the rate in which plants could colonize dry land massively increased in speed, coupled to the larger, thanks to wood, and more efficient, thanks to leaves. The physical impact of these plants on the environment changes relatively quickly. So these first land plants appear to be kind of paleogeographically very widespread, very generalists, and this is in large part due to the these very kind of novel features. Let's try and get a sense of what these changes look like. Let's start looking at the roots. So here we've got Rhynie from Rhynie Chert, and we can see it's tiny little roots here, and they're barely kind of novel extensions on the plant itself. But then here we've got Cairo, New York. This is, I think, 386 million years. You can see paleobotanists for scale. Like, these are massive. There's like 30 meters of rooting structures. You can see some of the aerial views here. Yeah, if you I should put a video in. There's a wonderful drone footage of the whole site. It's absolutely amazing. It's massive, massive trunks. Now, these roots are very, very important, doing many things, including stabilizing sediment, protecting from the land masses from erosion and rains from rivers. And here we get a sense of what happens when things go wrong. So when deforestation, you lose all that solidification of the sediment and you get massive changes happening. So, you know, when these mature forests are cut down, the water runs very quickly and often takes sediment with it, and you do get catastrophic landslides. But once you get to late Devonian and the modern vascular plants, we have these rooting structures that can stabilize. Now, this has many effects. So it includes having a lot more organic carbon that can be buried within the sedimentary rock. But it also changes the continental weathering regime. Ed gave a wonderful talk previously about this. So one of the other interesting things about it is that these very early plants had carbon to phosphorus ratios that were about 200 times more than is alive today, well, compared to modern marine biological systems, sorry, which means that for a given weathering flux, you have a lot more carbon being buried. Also, with land plants, you get changes in the hydrological cycle. So things like water cycling and rainfall, because more trees mean more rainfall through the taking in then evaporation of water, which then leads to rain elsewhere. But of course, a really big impact of land plants is on atmospheric oxygen. So today, land plants take up, produce about 50% of the oxygen, which is, the rest is all phytoplankton, which is a huge, huge amount. So to get an idea of how these processes complete, there's a very nice model here. This is kind of showing the kind of origins of land plants, then what happens if you have slightly earlier land plants in terms of oxygen levels, and what happens if you build in different sorts of weathering and different sorts of carbon and phosphorus ratios kind of consistently or as peak fluxes. So we've seen how the colonization of land and subsequent development of modern vascular plants has a really fundamental effect on the biosphere through increases in oxygen and changing the carbon and water cycles. All these different abiotic links link to an increase in ecosystem complexity. So there's now a very fundamental connection between what's going on in land and what's going on in the water that wasn't present previously. So things like massive phosphorus fluxes have been shown to really drive phytoplankton diversifications in the Meso and Cenozoic. But taking a bit of a step back and thinking about ecological stability. So I started my PhD here with Nick Butterfield and saw many variations on this. And it always perturbed me. And it took me a lot, it was only really preparing this talk that I worked out why it perturbed me. And it's this bit here. So we've got kind of time, animals evolving. We've got essentially the biomass or the morphological diversity, and it increases massively with animals. The length of time species live for decreases rapidly, and he always described this as stability. But it's kind of weird to think about it that way because these systems are not stable in the same way modern ecosystems are. And I think it's much more reasonable to talk about it in terms of you have simple ecosystems, but they're not really able to self-correct in the way that modern ecosystems can. They don't function in the same way. The reason they're stable is beyond their control. It's about abiotic factors. It's about what's going on under the earth. It's not about what the biology is doing. But once you get animals, that's different. The biology, the animals, and land plants, and all the rest of it have the capabilities to adapt and potentially stabilize their own systems. So, to summarize, I've been talking about ecological complexity through deep time. Start off with the origins of life on Earth, very much knowing that we need a very stable environment with widespread life in order to persist for 500 million years until we start seeing kind of cyanobacteria that can produce oxygen. These are helpful chemicals, is one way of phrasing it. But then when we get animals, things start to get very exciting because they start to be able to modify their physical environment beyond their lifetime, making it much, much easier for them and other animals to survive. The final transition I discussed was the evolution of these wonderful land plants, which is again almost doubling the amount of ecological complexity and linking it all together between land and sea. So yes, thank you for listening. I'd love to take questions.